Good afternoon one and all. So, here we are in our second session from our fourth chapter of PGT, that is cells and tissues. So far we have discussed about cell theory, different types and shapes of the cells, and we have begun with the prokaryotic cell. In prokaryotic cell we have clearly understood yesterday and we have discussed that the outermost layer of a prokaryotic cell, outermost covering of a prokaryotic cell is what? Cell envelope. Cell envelope is 3 layered, okay. Cell envelope is 3 layered, wherein outermost layer is what? Glycocalyx. Next to the glycocalyx we have cell wall and then we have cell membrane, okay. Glycocalyx, based on its composition, if it is tightly arranged, this is called as what? Capsule. If it is loosely arranged, this is called as what? Slime layer. Okay. And cell wall is made up of peptidoglycan. Cell wall is made up of peptidoglycan. And cell membrane will have some invaginations or inclusions towards the cytoplasm. Those invaginations can be in the form of vesicles, it can be in the form of tubules, it can be in the form of lamellae. These inclusions towards the cytoplasm from cell membrane are called as mesosomes. These mesosomes will do multiple functions like cell wall formation, DNA replication, daughter cell formation, respiration, secretion and increase in surface area. Amongst all this, the major function is respiration. That is the reason mesosomes of a prokaryotic cell will be equivalent to mitochondria of a eukaryotic cell. So, this is all about mesosomes. Such inclusions of cell membrane, these are the inclusions, right ma? These inclusions of cell membrane in blue-green algae or cyanobacteria, those inclusions are called as chromatophores. They are said to be chromatophores. Which perceive the sunlight and conducts the photosynthesis. This and all we have discussed yesterday. Now let us look into the cytoplasm. Inside the cytoplasm of bacteria, you have 2 types of genetic material, okay. One is called as genetic material in bacteria. Take down ma parallelly, genetic material in bacteria. It has 2 types of genetic material. One is said to be genomic DNA or chromosomal DNA, okay. Genomic DNA or chromosomal DNA. Second can be non-genomic DNA or extra chromosomal DNA or plasmid DNA. Fine. First, genomic DNA or chromosomal DNA, here if you take, this will look into all the metabolic activities of the bacteria, and genomic DNA might be or non-genomic extra chromosomal or plasmid DNA might be. Both of them are double-stranded circular DNA. What mode of DNA is present na? Double-stranded circular DNA. Fine. And this will look into what? Metabolic activities of the cell. Whereas non-genomic DNA, extra chromosomal DNA or plasmid DNA, this will be looking after 3 major functions in providing specific phenotypic character to bacteria, okay, in providing specific phenotypic character to the bacteria. Second is resistance to antibiotics. Resistance to antibiotics. Okay. And third will be promoting the sexual recombination, that is conjugation. Okay. Promoting sexual recombination, that is conjugation. Fine, ma? So, here we have inside the cytoplasm of a prokaryotic cell, two modes of genetic material are present. One is genomic DNA and the other is plasmid DNA. Both the genomic DNA and plasmid DNA are double stranded circular DNA. Genomic DNA will look into the metabolic activities inside the cell, whereas plasmid DNA will give specific phenotypic character to the bacteria. It provides resistance to antibiotics and sexual recombination, that is conjugation, is conducted. How that will happen is, those plasmid DNA which has F factor, are said to be F plus strain of bacteria. So, those bacteria which has F plus strain will come in close proximity with those bacteria which does not have F plus strain. Okay. So, this is said to be what? F minus, and one which has is called as F plus strain. Fine. This does not have. Okay. These two bacteria will come into close proximity by true pili or sex pili or conjugation tube. So, a conjugation tube, true pili or sex pili is formed. True pili, sex pili or conjugation tube is formed. Fine. True pili, sex pili or conjugation tube is formed in between two different strains of bacteria, that is F plus and F minus. As such, the F factor from the F plus strain of bacteria is given to the F minus strain, and the products will be F plus and F plus. Fine. And there is one more mode of conjugation, that is HFR to F minus. HFR to F minus strain. So, here if you take two different strains of bacteria, F factor, if it is present in the genomic DNA rather than the plasmid DNA, then it is called as HFR, okay, wherein F factor is present in the genomic DNA. F factor present in genomic DNA. Fine. This F factor is being transmitted from genomic DNA of HFR strain to plasmid DNA of F minus strain, like this. Okay. So, this will give out the products, those are HFR and F plus strain. Fine, got it. So, these are the uses of what? Plasmid DNA. Fine. So, genetic material of bacteria, it has two modes of genetic material. One is genomic or chromosomal DNA, and the second is non-genomic extra chromosomal DNA and plasmid DNA. So, here this both of them are what? Double stranded circular DNA only. Genomic DNA will conduct the metabolic activities, whereas plasmid DNA will look into the specific phenotypic character, resistance to antibiotics and sexual recombination, that is conjugation, in between F plus strain to F minus strain and HFR to F minus strain. Now, we will also look into the non-membrane bound organelles, okay, then we will go for gram staining. Yeah, non-membrane bound organelles in the bacteria. What are the non-membrane bound organelles that we see is ribosomes and inclusion bodies. Inclusion bodies. These are what? non-membrane bound organelles that are observed in the prokaryotic cell. Non-membrane bound organelles. So here if you see, ribosomes are of what type? 70S ribosomes are identified. Larger subunit and smaller subunit, if you see, larger subunit is 50S, smaller subunit will be 30S. Fine. These are the ribosomes. Ribosomes main function is what? Protein synthesis. Protein synthesis. And only in the prokaryotic cell or bacteria we can observe something called as polysome or polyribosome condition. Polyribosome condition, which means a single mRNA strand will have many ribosomes present over it. Single mRNA strand will have many ribosomes present over it, which synthesize multiple proteins and gives rise to polycistronic expression. Polycistronic expression. Fine, okay. And inclusion bodies are nothing but these bodies will store reserve food material, okay. Inclusion bodies will store reserve food material. Reserve food material inside the bacterial cell. Fine. So, this reserve food material can be glycogen granules, phosphate granules, cyanophycean granules. Fine. So, these are the reserve food material inclusion bodies. There is no membrane bound organelle that is identified inside the bacteria. Okay. Now, we will be looking into the motile and non-motile structures in bacteria. What are the motile and non-motile structures? Okay. Motile and non-motile structures. Fine. Motile structures include only flagella. Flagella is the only motile structure that is observed in the bacteria. Non-motile structures include pili and fimbriae. Fine. Flagella will originate from the cell membrane of the bacteria. It will originate from cell membrane of the bacteria, and flagella will have three parts in it. One is basal body, hook and filament. Okay. Basal body, hook and filament. Basal body has microtubular arrangement same as that of centriole, and hook and filament also. So, complete flagella will show 9+0 microtubular arrangement, which means at the center there is a proteinaceous hub. To this proteinaceous hub 9 radial spokes are arising. To each radial spoke 3 microtubules will be present, okay, like this. And how flagella will be moving? Like this, right. So, it is of what arrangement? 9+0 microtubular arrangement, both basal body, hook and filament. And what is the protein that is present inside the prokaryotic flagella? Flagellin. Flagellin is the protein that is present inside the prokaryotic flagella. Now, when it comes to the non-motile structures, those are pili and fimbriae. Pili are nothing but tubular extensions from the cell wall. which helps in attachment of this bacteria to the substratum. Okay. Pili are tubular extensions from the cell membrane. This will help the bacteria to attach onto the substratum. Next is fimbriae. Fimbriae are nothing but bristle-like structures sprouting out of the cell. Okay. This also helps in what? Attachment to the substratum. Fimbriae are bristle-like structures sprouting out of the cell. Clear? Pili is also called as sex pili or true pili because its function's major role is conjugation, promoting conjugation or forming out conjugation tube. This is all about the structural description of a prokaryotic cell. Now we will look into different shapes and sizes of the cells. Prokaryotic cell and different sizes of the cell. Prokaryotic cell based on different shapes. If the shape is spherical, it is what? Cocci, and singular form is coccus. Okay. If the bacteria is rod shaped, then it is bacilli, singular form is bacillus. Fine. Spirillum, it is spiral shape. Okay. And plural form is spirilla. Fine. Vibrio is comma shaped. Fine. And singular form is what? Vibrium. Plural is vibrio. Okay. And if you see different sizes of the cell, this is a typical eukaryotic cell, and eukaryotic cell whose size is 10 to 20 micrometers in diameter, and bacteria is 1 to 2 micrometers in diameter. Pleuropneumonia like organisms, it is of 0.1 micrometers in diameter, and viruses are 0.02 to 0.2 micrometers in diameters. Of this, which is the smallest cell is pleuropneumonia like organisms, because these are the examples of mycoplasma, which is the smallest living cell. Okay ma? And then Gram staining. Gram staining is a technique which is developed by Christian Gram. Where in this staining procedure, okay, first the bacterial smear is being dyed with crystal violet. So here it is given clearly, bacterial smear is first dyed with what? Crystal violet, and it will show what color? Purple color. Then a mordant is added, which is iodine, and then it is washed out with alcohol. Further one more stain is added, that is safranin. Few bacteria are still retaining the color of purple, whereas few bacteria has shown pink or orange color because of the staining of safranin. Okay. Those which retained the color are said to be gram positive bacteria. Those which could not retain the color are said to be gram negative bacteria. Fine. So here if you see gram positive bacteria, why is it able to retain the stain? It is because gram positive bacteria will have thick cell wall layer which is made up of peptidoglycan. For example, take a soft silk cloth and a thick cotton cloth. Okay. Both of them you dip it in the water, okay, and wash it out. Whose color is easily gone? Silk cloth color will be easily gone, whereas cotton will absorb the color. It is because tight composition of the fibers inside the cotton, right. Same the case in peptidoglycan of gram positive bacteria, it is tightly arranged. Okay, cell wall is tightly arranged with peptidoglycan, and there is less composition of lipopolysaccharides. Okay, and this also has techoic acids and murin composition. So this composition will make it thick and tough, where it can retain the strain for long run. Whereas this, if you see gram negative bacteria, where the stain is easily dissolved because peptidoglycan is very less. Just here it is 80% of peptidoglycan or 80% of murin is present. Here it is just 10% of peptidoglycan is present. Whereas 80% it is composed of lipopolysaccharides. Techoic acids are absent and there is no murin. That is why it cannot retain the stain. It can easily absorb any new stain. Okay. So this is all about the experiment. So what I told you, peptidoglycan is around 80% in gram positive bacteria. In gram negative bacteria, most of it is composed of what? Lipopolysaccharides only, and peptidoglycan is just 10%. And gram positive bacteria is rich in what? Techoic acids and murin. Best example for gram positive bacteria includes Bacillus, Corynebacterium, Clostridium. Fine. Gram negative bacteria includes Salmonella typhi, E. coli. E. coli is also a gram negative bacteria. Azotobacter or Acetobacter. These are the best examples of gram negative bacteria. So understood, right? The staining procedure and gram positive, gram negative bacteria. Now we will look into the clear differences between prokaryotic cell and eukaryotic cell. In our next class, we will look into the complete details of a plant cell and animal cell. So now if you see prokaryotic cell and eukaryotic cell in detail, first, nucleus is absent in a prokaryotic cell because in a cell genetic material is completely not scattered, it is present at one position, which is called as what? Nucleoid. Whereas in eukaryotic cell, true nucleus is present with the nuclear membrane, and here nuclear membrane is absent, nuclear membrane is present in a eukaryotic cell. DNA type is circular and naked DNA. Here DNA has linear DNA with histone proteins. Okay, DNA which is negatively charged is packed with positively charged histone proteins. Membrane bound organelles are absent. Here membrane bound organelles are present. Ribosomes are 70S, where larger subunit is 50S and smaller subunit is 30S. Here ribosome is 80S, where larger subunit is 60S and smaller subunit is 40S. Cell size is 1 to 10 micrometers in diameter, and here cell size is large, that is 10 to 100 micrometers in diameter. And then cell division, here if you see prokaryotic cell, binary fission, eukaryotic cell, mitosis and meiosis. Cell wall composition, here in prokaryotic cell it is made up of peptidoglycan, eukaryotic cell it is made up of cellulose, chitin and fungi, and absence of cell wall is seen in animal cells. Examples include bacteria and archaea. Here in eukaryotes, examples include plants, animals, fungi and protists. Cytoskeleton is absent or rudimentary. There is no proper cytoskeleton in prokaryotic cell, but here in eukaryote cytoskeleton is present, which has proteinaceous filaments like microtubules, microfilaments and intermediary filaments. Plasmids are most commonly observed. Here plasmids are not observed. Mode of reproduction is asexual reproduction or binary fission over here. Here both asexual and sexual reproduction is observed. Here sexual recombination is seen in prokaryotic cell like conjugation, transformation and transduction. Gas vacuoles are present. Here gas vacuoles are absent in a eukaryotic cell. So these are the fundamental differences between prokaryotic cell and eukaryotic cell. In our coming class, we are going to learn in detail about the eukaryotic cell, plant cell and animal cell. We are going to fragment the concepts into non-membrane bound organelles, single membrane bound organelles highlighting the endomembrane system, and double membrane bound organelles. So, that is all for today's session, and thank you.